afternoon, me again, and I am speaking for the actually the Pediatric Hematology Oncology team, which studied blood. Okay. So we are in the Hong Kong Children's Hospital, and we are actually touching a disease known as childhood acute leukemia. Actually, it is the most common malignancy in children, representing over 30% of the cases of all cases of cancer. The incidence of childhood leukemia is about 40 to 50 per million children, and in Hong Kong we have 1.1 million pediatric population, so we will have a 60 new cases per year, all recruited in Hong Kong Children's Hospital now. Okay, so that I moved there for my research. So the standard of care of childhood leukemia remains unchanged in the last few decades, I can say, and based on intensive chemotherapy plus or minus hematopoietic stem cell transplant, the blood stem cell transplant. Okay, some of you may have listened to my lecture during our delivery in CUHK. So with this regimen, actually, you can see that the overall survival of childhood leukemia is improving substantially from approximately to less than 20% in the 1960s to over 90% nowadays. Okay, so very good improvement without the change of any drugs. Okay, however, there are some difficult cases. We have difficult cases that have disease recurrence after chemotherapy. Okay, they become resistant to chemotherapy, and their survival is extremely poor, less than 40% survival for those who has disease recurrence. So we need to do something, okay? And it is driven by research. So in our team, actually has 3 academic staff, 2 Professor C.K. Lee. It's actually the king of leukemia, I can say, in Hong Kong, who lead the leukemia team in the Hong Kong Children's Hospital. And Dr. Alex Leung, both of them are working on the clinical hematology for clinical research. And myself is working on, I'm graduated as a basic scientist and work for hematology, so I'm doing translational hematology research, I can say, okay? So this is our team structure, currently 15 people in my lab, okay? Everyone contributed to very significantly to my projects. So my lab is situated in the Hong Kong Children's Hospital. As Renee has mentioned, we have 2 towers. One is research, and one is the clinical block that manage patients. So we are interconnected. For everything the research floor fill up, we translate into the management of patients. And then they will have new clinical questions and then answered by the research floor. This is the idea of the Hong Kong Children's Hospital. Very nice hospital with a great sea view to Victoria Harbour. So what makes our team distinct? We are the only team in Hong Kong dedicated to drive the research excellence of childhood leukemia. We are the only team doing childhood leukemia research in Hong Kong. Okay, therefore, we have exclusive patient recruitment. Since I started leukemia research in 2013, okay, 9 years ago, we have already banked 450 bone marrow samples from leukemia patients. Very big cohort indeed. Or you can foresee we have 60 cases per year. So 10 years effort, and we build a cohort with a clinical annotator. We have survival data. So many of the research can be done. Okay, we have proven track record. #1 journal, #2 journal in hematology. We have published on that. We also have a very strong collaborative network, as mentioned by Ginny and Renee. Collaboration is extremely important nowadays. Okay, we have collaboration with UK and the biggest children's hospital in the US. Okay, St. Jude Children's Hospital. We have the biggest children's hospital in China. Okay, Shanghai Jiao Tong Dashi. then we are touching on the whole population of leukemization in China. We are talking about 9,000 patients nowadays. Okay, we have that data. And then the Macau, so we are interconnected with the world. Okay. So some of my research interests, since I am embarking a PhD at the same department here, date back to 2006, my supervisor here gave me a topic to investigate the mechanism of blood stem cell home. That is very important for stem cell transplant. You are transplanting your stem cell in your peripheral blood, but the home of the stem cell is the bone marrow. Okay, so how they recognize their roots to the bone marrow, it is my research interest. So in this department, we are connected with the ONG department to collect umbilical cord blood from the newborn neonate. And then we isolate the hematopoietic stem cells, very routine work nowadays, okay? And then do a cell culture, cell migration assay, and then transplanted it to a immunodeficient mice, mimicking a stem cell transplantation in animals. And then identify how the stem cell find their way to the bone marrow, okay? We have published quite a number of papers on this. My first paper in this department is identify the CD9 protein that is involving in stem cell homing, which is published in Blood. Okay, and then recently, my colleague Kathy Chen has published 2 papers in Blood Advance and also in Journal of Leukocyte Biology, identify some other protein that is important for stem cell homing. So what is the outcome? We want to utilize the result to improve the stem cell transplantation successfulness. Okay, so we are always doing a research for reason. Why? We want to look at the mechanism because we want to improve the patient outcome. Okay, we need to understand the basics. So since I finished my PhD, I run on to my postdoc training at the same department. Okay, so met my mentor, Professor C.K. Lee, and then touch about leukemia. Okay, he said to me, why don't you measure CD9 in the leukemia sample? And then I said, okay. Then they gave me 10 samples. I found that 7 of them are positive. Okay, this made me very exciting work. And I started my leukemia research in 2014. Okay, 8 years effort. So my first paper on leukemia is published 2 years ago. It takes 6 years to publish my first paper in leukemia. Okay, it's a long process. And then identify the same gene, identify stem cell is important for leukemia progression. Okay, when we block CD9 using a therapeutic antibody, I can say it's a monoclonal antibody. You can say the leukemia are all gone. Okay, this is a commercial available antibody that I'm purchasing from PD, okay? But now we have our own antibody, fully human antibody with an engineering that show very great promise in the future. And we are planning to do primate study in the next, actually we'll be doing very soon. Okay, we want to move the CD9 antibody into the clinical trial. Okay. Second, CD9 CAR T cells, a very hot topic. CAR T cell probably is the most powerful tools nowadays for relapse or refractory leukemia. Then we generate the CAR T. Actually, this is a T cell from my blood. Okay, we make it into a weapon. Okay, with a red fluorescent tag CAR T cell. The leukemic cells are labeled in green. You can see the T cell, CAR T cell, chase the leukemic cell, and then they die. Okay, this is the powerful CAR T cell. Okay, we are doing research on CAR T cell, how to improve the weapon. Okay, this is the main research angle we are looking at. We are also having CD9 small molecule inhibitor. Actually, indeed, I have a postdoc who is graduated from the chemistry department. Okay, the team multidisciplinary. Each one have a specialty, expertise, and then we successfully developed the first in class, actually the first in the world, CD19 in small molecule inhibitor, which we are undergone patent protection. Okay, so all of these are hope for the student, hope for the patient, sorry, and then we are doing it for those who has no further treatment option. Okay, you can consider these are future drugs that may be driven a cure in leukemia. Immunome of my childhood leukemia, we talk about genome, we talk about metabolome, many big data, we understand the whole spectrum genomics in leukemia. And then leukemia, blood cancer, actually indeed is not easy. It's composed of this one leukemia, we call it acute lymphoblastic leukemia. Recently, a paper published in Nature Genetics identified it has 23 subtypes. Okay, even you call it a leukemia, there are 23 different subtypes of this leukemia. Interleukemia heterogeneity is the thing why some patients respond to chemotherapy, why the others don't respond, because of the genetics. Okay, because of the genes that control the response to chemotherapy. So in every cases we collected for leukemia in the Hong Kong Children's Hospital, we send for RNA sequencing. So we are building a big data set. Okay, nowadays we have already housing 150 RNA sequencing data for the leukemia case. Okay, even within an individual patient, okay, when you look at the population of leukemia, it is different because the bulk leukemia is consisting of 1 million of cells, that the cells may be different from each other. So these are intraleukemia heterogeneity. And then we are now planning, actually we will start very soon, we have ordered the reagents, and we will start doing single cell RNA sequencing to understand not just the leukemia, okay, but understand their partner in the bone marrow. For example, the infiltrated T cells, the infiltrated B cells. What are they doing? What are they talking with the leukemia? Are there any important within it? This is the very basic research, right? We have very immediate research of very immediate impact. So in 3 to 4 years ago, I recruited a PhD from Zhongshan Daihao. It's a Sun Yat-sen University. Very talented. This one is called Wang Han. Okay, it's now my postdoc already. So since her arrival, we started a program that is called precision medicine for those untreatable leukemia. Actually, when we have a case that the doctor say this is untreatable, they send us the bone marrow, and then we isolate the leukemic cells, and then we profile the drug sensitivity within a very high throughput assay, and use only 3 days to report to the doctor. The patient is likely to benefit from each drug. Okay, and then this is the first case in Children's Hospital we treated. Okay, he is defined as untreatable at that moment. He developed relapsed leukemia after stem cell transplantation. Usually, this patient will die after 2 or 3 months palliative care. Okay, but we do the drug profiling, identify a good drug, and you can see after a week, the leukemia are gone. And the patient actually have a 2.5-year survival now. Okay, very promising. And then the article is recently just published online. And then my PhD student, Wang Han, had actually got the very prestigious award in the International Society of Pediatric Oncology. Probably she is the first Chinese getting this award. Okay, this is how we train the student of our hematology team. So this is a leukemia survivor. Okay, actually in my lab, actually I recruited multiple students. That some of them are actually leukemia survivor, I can say. Okay, please do consider joining our team and work for the kids. Thank you very much.